next speaker is also from Zurich, Lukas Roth. Thanks, Lukas.
Yeah, thank you. So after those 3 talks, I think everything is said about phenotyping, and you have the chance to repeat with me, the basis of it in this talk. I will start with what motivates us. We have heard that yield is not increasing anymore, and we have issues with increasing the genetic gain. That's the case for Switzerland, but it's also for France and for several other countries in Europe. And an assumption is that adverse climatic conditions are causing this. And I do research in Switzerland, so next thing I have to prove is that actually we have G/E in Switzerland, which I did here. So these are data from variety testing from 4 years in Switzerland. The blue dots are the sites. It's in the wheat belt, so the rest of the area is mountainous areas where you cannot grow winter wheat. And you see that for grain yield here, we have large components related to the environment. Then we have this genotype effect here, but we also have genotype-environment interactions, which are almost as strong as the genotype effect. For heading data, it's different, almost no interaction. Here, plant height is huge genetic effect, most probably caused by reduced height genes. And then for protein content, grain protein content, again, the same as for grain yield. So we have a component which is related to G/E. We should investigate G/E to further improve our breeding programs. And how we do that, usually, I borrowed this graph from Hans-Peter, and I'm misusing a little bit for my purpose, so I hope he does not mind. Complaints in the break. So what we take is we take a lot of genotypes and we use marker data, for example, to characterize them. We take a lot of environments and we also characterize the environments with environmental covariates. And then hopefully we grow phenotypes on those genotypes in these environments. And if we then take just the marker data for our predictions and do not model G/E, that's classical genomic predictions, but we can become better, include the environments. We have seen examples. And what we can also do, we have seen an example from Daniela, we could include phenotyping data. So other data than we are interested in, and not the target rate yield, but for example, heading data. Now this calls for models that can handle such missing modality at inference, because for a new environment, obviously we do not have phenotyping data, and prominent examples are transformer-based deep learning models that come from such use cases where one modality or another can be used for doing prediction. Now, this graph helps me a lot to understand how those models work, but I have an issue with it when I'm in Zurich. And we have this very nice, typical ETH platform, very accurate, very expensive, and it exists only in one location here. So I cannot move it to another place. I cannot repeat measurements in other environments. I just have that one environment. And this is the same for the breeding program in Switzerland. We have a mid-sized breeding program for winter wheat, and up to the F5, 6 generation, those varieties just exist in one location because there are not enough seeds and also not enough money to replicate those seeds at other places. So our situation is actually more like that. So we still have a lot of genotypes. We can characterize them beautiful, but well, just 1 or 2 environments. And obviously we cannot grow the genotypes in more than those 2 environments. So what can we do? What we can do is phenotyping. We can take a drone, we can monitor the plants as they grow over time, and we can repeat our measurements, so we can now describe the phenotype of the plants at time point 1, 2, 3, 4, 5, and so on. Depends on how many times you measure on the field. Now, the nice thing about environments is that we can also characterize environments very cheaply. So we can monitor precipitation events, temperature, so we can also characterize the difference between those time points in the environment. And then we can take this concept of modeling genotype environment interactions out of the black, I sometimes say, or out of the abstract, and actually model short-term responses, physiological responses to the environment. So we can try to describe what happens to this phenotype between those time points in relation to what happened to the environment between those two points in time. And the hypothesis is then, if we do that, we can extract such parameters that are genotype-specific, and together with genetic main effects, we can use those to train a predictor that can model the performance of genotypes in new environments and new years that we have not seen yet. So that's the hypothesis why high-throughput phenotyping is useful. How we do high-throughput phenotyping, you have seen in Olivia's talk. We have this platform, a rope-suspended system here with a camera. This is the new sensor head with 13 cameras. This is the old one with just one RGB camera. And we have a field which is large enough to host around 300 winter wheat genotypes with 2 replications. We do also other measurements. If we need to be more close, we can also track individual leaf organ growth, or for thermography, it turned out to be useful to be faster than actually this platform here in the middle is, because you need to integrate your measurements in a very short time to have a reliable signal. No matter what sensor you use, you end up with a bunch of data you need to process, and here comes feature extraction into play. So we extract plant heights here, for example, canopy cover. We can also use with plain image as input, and we recently went into also reconstructing 3D architectures of canopies and take that as input for our models. Now, independent on how you extract the features, you end up with time series of traits that develop over time. And this is an issue because, well, that's a time series, and yield is a scalar, end of the season, just one value. So we need to somehow model the dynamic to extract something which we then can correlate or use as a predictor for yield. And I thought a lot about that, and in the end turned out that you can basically do 3 things. You can focus on the time axis and extract turning points. So as Olivia showed, start or stop of the stem elongation. You can focus on the y-axis, but then you need a defined point in time where you say, well, after they stopped stem elongation, I extract the final height here, for example. Or you can go in the direction that you try to model the dynamic here in between in relation to environmental covariates. We also investigated how many times you have to measure to answer that question for those traits. Twice a week, and you can rest on weekends and public holidays. That's in that paper here. So it's citable for PhDs who need arguments for their supervisor. So once we have extracted those parameters, the question is that just something, or does it have a genetic base? And for that, we worked with the large genotype panel that was grown for years on our field, 350 genotypes sequenced, or not sequenced, characterized with a breeder's chip, SNP chip. We measured the height increase over time, as already described, and then modeled the dynamic. And first, we did what I said on the time axis, so extracted turning points, start and stop, and final height here on the y-axis. There is a strong clustering based on the breeding origin of those genotypes. So we, for example, realized that in rather continental areas, they start early in the stem elongation, and in rather Mediterranean areas here, France and Great Britain, they have a delayed start of the stem elongation. Similar patterns for final height, other clusters for the stop of stem elongation, and then if we go into modeling the response to temperature, again, new patterns that arise. Now, this genotype panel has a very diverse background, so that could be all population structure. So we questions whether we can also do GWAS and actually find market rate associations for those traits, and it turned out yes, many. So we found here for all the traits we investigated a lot of market rate associations, and most of them were not shared. So here on the left, the ones that were shared amongst traits, and here the ones that were independent. So we could conclude that, well, those traits have a quantitative inheritance and a large degree of freedom or independence among loci. So one can say, well, with such an approach, we can model short-term responses, and it has a genetic base, what we extract. So the implications for breeding and predictive modeling is now that we can extract those traits and we can train a predictor, as I described here. And that's the next step we did. Here the question mark is resolved. We get a prediction accuracy which is moderate, does not outperform other models. It has to be said that this prediction accuracy is on an unseen breeding generation measured on the field. So it was trained on 5 years of variety testing and predicted on a field site of an F7 generation panel. But apart from the prediction accuracy, what is interesting about such approaches, to give Daniela some hope, is you can interpret it. So if you do not outperform the baseline, but you have a predictor, such as, for example, here, a partial least square regressor, which indicates the importance of different features, you can use those features and compare it with what we did here with the breeder selection. So we realized breeders mainly select for early vigor here, number of tillers, but our model also suggested to go for a base temperature of growth here, for example, which is not in a one-to-one relation to protein content. So it could make sense to select for that, and because it reduces protein content, but not one-to-one to yield, and then compensate for this reduction in protein content with another trait and combine them. This is what we suggested as ideotypes that are based on such phenotyping. And we did that here for wheat. So actually we said, well, one could increase the base temperature of growth to increase yield, but at the same time, if one selects for an early start of the stem elongation, it could compensate for the reduction of protein content that comes with this increase in yield here. And the same for soybean. Good. Now, as an outlook, what I presented here and also all the speakers presented is basically what Fred and colleagues suggested in that paper. So to do this stepwise approach of extracting feature, modeling the dynamic, bring that dynamic somehow in relation to a target rate, and then in the end extract an ideotype. And I have to make a lot of decisions on that way. Which trait do I measure? Which dynamic parameters do I have to extract? In which time of the season do I measure, and so on. So I would be interested in an approach that could do that all at once. So that takes image time series as input, embeds what it sees in this image time series, uses that embedding to then predict target trait, and then I could ask the algorithm what it was looking at instead of doing that process here. So we are working at the moment on such models. I can present you here a preliminary result. So that's a two-branched model. Takes in one branch the image time series as input and the embedding of those image time series. And then the model is forced to do the same just based on market data and environmental covariates. So those two states have to be the same because and the model does not know which one is then taken to the next step to predict the final target rate yield. And this then can simulate kind of a G/E contribution, and then the pure genotype contribution comes from the market data. Now, you may have to validate such an approach, of course, we have to go outside the platform here. So I use similar varieties that were measured in variety testing in Switzerland and in this public multi-environment trial data from IPK from Germany to have an independent test set. Now, a small déjà vu. In green are the baselines, and in red is the performance of that model. It does not outperform that model again, but I can visualize what the model actually has learned. So what you see here is this latent space that the model thinks how a plant has to develop and that it uses as input to predict the target rate. And now I can try to cluster those trajectories based on, for example, which genotype is highly sensitive to yield and which one is rather stable for environmental conditions. And again, try to formulate ideotypes that describe, for a certain target population of environment, a stable or a rather dynamic genotype. So that can be hopefully continued in the next presentation. If you now think that's nice that those guys can do that, but we don't have the data, that was true until last week. But then my PhD published a data set consisting of 6 years of data that we collected with that platform. So it has all the images we collected for over 300 genotypes a year for 6 years. It also has genetic marker data associated with it, based on this public market data from the Gabi panel. There is an addition where you have to ask us whether you can use it or not, but that would also be available in a collaboration. If you don't like to work with images, but like to have longitudinal data, we also extracted from the image canopy covered plant height and senescence rating and weed head counts. So you could also work with those. And yeah, last slide. And because it's always an issue to compare your performance with others, what we suggest is a training test split that you can follow to be able to compare your performance with the baseline that we also published with that data set. So this is the link you all hopefully all have been waiting for to access the data and download it. And with that, I'd like to thank you for your attention.